In the quest to slow biological aging and protect DNA, both vitamin D and omega-3 fatty acids have attracted considerable scientific attention. These two nutrients are often studied side by side because they share anti-inflammatory and antioxidant properties that could theoretically help preserve telomere length, a marker of cellular aging. However, when examined in certain clinical trials, notably those exploring lifestyle and supplement interventions, vitamin D has consistently shown a more pronounced benefit in telomere protection compared to omega-3s, even when both were administered under similar conditions. This difference raises important questions about the mechanisms behind their effects and why one nutrient might offer more direct cellular protection than the other. One of the key reasons vitamin D appears to outperform omega-3s in telomere preservation may lie in its hormone-like role in cellular regulation. Vitamin D is not just a nutrient, but a secosteroid hormone that influences the expression of hundreds of genes, many of which are involved in DNA repair, cell cycle regulation, and inflammation. Vitamin D receptors, VDRs, are found in virtually every cell in the body, and when activated, they modulate processes directly tied to telomere dynamics. For example, vitamin D may reduce oxidative stress and chronic inflammation, two of the primary accelerators of telomere shortening, through direct genomic signaling, which includes enhancing antioxidant enzyme activity and repressing inflammatory cytokine production. In contrast, while omega-3 fatty acids like EPA and DHA also reduce inflammation and oxidative damage, they tend to work through indirect pathways, such as being incorporated into cell membranes and altering eicosanoid signaling cascades. Their effects may be more systemic and less targeted toward genomic preservation than those of vitamin D. This broader, but perhaps less focused action may explain why omega-3s did not consistently show telomere lengthening effects in some studies, even though they still provided cardiovascular and metabolic benefits. Another factor is the specificity of biological targets. Telomere length is maintained not just by reducing damage, but also by supporting the activity of telomerase, the enzyme responsible for rebuilding telomeres. Vitamin D has been shown to upregulate telomerase expression in certain cell types, such as immune cells, which could directly influence telomere length maintenance. Omega-3s have not demonstrated the same clear effect on telomerase activity in human trials, possibly limiting their role in telomere-specific repair. Bioavailability and baseline status may also play a role in observed differences. Many participants in vitamin D studies start with suboptimal levels, especially in populations with low sun exposure or darker skin pigmentation. When supplementation corrects a deficiency, the resulting improvements in immune regulation and cell function can be substantial, including measurable changes in telomere dynamics. Omega-3 levels, while also often low in Western diets, may not reach a similarly profound threshold effect from supplementation unless the baseline deficiency is severe or the dose is extremely high. Additionally, omega-3s may be more effective in people with specific inflammatory conditions, whereas vitamin D seems to offer broader protective benefits across a wider range of health statuses. It's also important to consider study duration and endpoint sensitivity. Telomere length changes slowly and is influenced by a wide range of factors, including stress, infection, and metabolic health. If the intervention period is too short or the population too heterogeneous, subtle benefits from omega-3s might be overshadowed or undetectable compared to vitamin D's more direct genomic effects. Furthermore, different methods of telomere measurement, such as qPCR versus flowfish, may vary in sensitivity, potentially influencing study outcomes in nuanced ways. Finally, the interaction between these nutrients themselves could be relevant. Some researchers speculate that vitamin D may actually enhance the effectiveness of omega-3s, or that both work better when combined with other antioxidants or micronutrients like magnesium or vitamin K2. However, when evaluated in isolation, vitamin D may simply have a more central role in maintaining the structural and functional integrity of DNA, particularly within immune and endothelial cells. In conclusion, while both vitamin D and omega-3 fatty acids offer significant health benefits, vitamin D appears to have a stronger and more direct influence on telomere preservation and DNA protection in certain trials. This difference likely stems from vitamin D's genomic signaling capacity, its ability to activate telomerase, and its systemic role in inflammation and cellular aging pathways. Omega-3s, though beneficial in many contexts, may exert their influence through broader metabolic pathways that do not target telomere dynamics as precisely. These findings underscore the complexity of nutritional science and suggest that not all anti-inflammatory nutrients have equal impact when it comes to cellular longevity, a critical distinction for those designing anti-aging strategies based on evidence-driven interventions.